Today we are going to study the duplex model of DNA. Duplex model of DNA. What is a duplex model? So duplex model means double helical structure. Double helical structure of DNA. Double helical structure of DNA, and this was first time proposed by Watson and Crick. First time proposed by Watson and Crick. Hence, it is also called as Watson-Crick double helical structure of DNA or Watson-Crick duplex model of DNA. And for that, they also awarded Nobel Prize for their brilliant study on the structure of the duplex model of DNA. In the previous class, just I have introduced about the polynucleotide chain. What is a polynucleotide chain and what is nucleotide? Polynucleotide chain. About polynucleotide chain you studied, and that to be single polynucleotide chain, that one you studied in the previous class. And the nucleotide, the word nucleotide means it is the composition of nitrogen base, next the pentose sugar, and the last one the phosphoric acid. All together called as a nucleotide. So means nitrogen base, the pentose sugar, and the phosphoric acid. And that you studied in the last. Once again I will remind you this one, the nitrogen base, and to this attached is what? The pentose sugar. To this it is attached the pentose sugar, and then here what? The phosphate. And this one complete is what? The nucleotide. One complete is a nucleotide. So that form the structural and functional unit of the DNA. What are nucleotides? Nucleotides are the functional unit of the DNA, and these nucleotides here held with one another or linked with one another, understood? And that is called as polynucleotide chain. So means here one more I can write here. For example, here it is A like this, pentose sugar and the phosphate. See, so it is linked. So this is also nucleotide and this is also nucleotide and linked with the phosphodiester bond between sugar and the sugar, and each phosphate molecule having two sugar phosphodiester bond, and thereby form many nucleotide formation. So like this here also you have studied in the previous class, and that is the guanine. So guanine, then the pentose sugar, and then here the phosphate, and also here is the phosphate. So like this here, the one side what the thymine, the adenine, the guanine, next one cytosine, and these all are arranged by formation of large number of nucleotide, and that form a polynucleotide. So means this is a one side the strand of the DNA. It is a one side strand of the DNA. So another side of the strand of DNA means what here? Now I have to connect here. So like this AT double bond A, and to this here once again the pentose sugar. See, pentose sugar and the phosphate. Correct. And similarly here A double bond T, and then pentose sugar, phosphate. So this is a phosphate, and here is also the phosphate. Similarly G triple bond C, and then pentose sugar like this. Here form a double strand. A double bond T, T double bond A, C triple bond G, and that form what here? The double stranded polynucleotide chain. double-stranded polynucleotide chain. In the previous class, you studied single-stranded polynucleotide chain. This is one single strand and this is one single strand. But here, what I did, both the strands here united by what here? The hydrogen bond. Understood? Both the strands, just like a ladder. See, just like a ladder. This ladder is made by the sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, like this. And thereafter, here these two, what are the strand connect by means of here what? Hydrogen bond, A double bond D, G double bond C, C triple bond G. So like this, they connected with the one another. So what I gave just now is the double-stranded polynucleotide chain, how they are arranged, what in a given the duplex model of what Watson and Crick. So same now you have to study here in one structure. This is a right-handed fashion structure of DNA molecule. It's the same structure already how I gave you it is in the previous class also. Say like this, if it is that A double bond T, T double bond A, C triple bond G, C triple bond G, G triple bond C. So this A double bond T, T double bond A, T double bond A double bond T. You can write in any fashion, in any way, but the pairing should be one furin, one pyrimidine. One furin, one pyrimidine, because here you know in the nitrogen base, the furin bases are the and pyrimidine bases are the furin bases adenine and thymine, and then pyrimidine bases are guanine and cytosine. That part also you studied in the previous class. So that is why the base pairing is fixed. Adenine never combine with the what? The cytosine or guanine. It always combine or pair with the thymine only, with how many hydrogen bond? 2 hydrogen bond. Similarly, T double bond. It's the vice versa. Then cytosine always combine with the guanine, and this cytosine never combine with thymine or the adenine. And that to be cytosine combine with the guanine with how many? The 3 hydrogen bond only, and that is a specific, and the vice versa. G triple bond C. Like this, in a DNA molecule, all these nitrogen base pair in any fashion, and so in this way, here you see the double-stranded DNA molecule formation taking place, and in this double stranded DNA molecule, here what that backbone is made by sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. This means the strand, the entire strand is made by the sugar and the phosphate, sugar and the phosphate, and the parallel, the opposite strands are connected by means of here the hydrogen bond in terms of nitrogen bases. So in this way, nitrogen base, pentose sugar and the phosphoric acid that form the functional unit of what DNA, and that is what you call as a nucleotide, and that form one polynucleotide chain. This is one polynucleotide chain, this is one polynucleotide chain, and this is also another polynucleotide chain. So thereby these both are the anti-parallel. So that is why the general characteristic feature of the duplex model of DNA, here the first one is the DNA strands, double strands are anti-parallel. The double stranded molecules are what? The anti-parallel. They are what? The anti-parallel, and so their chain, the single stranded chain, and they both are the anti-parallel. So anti-parallel means what? If it is here is a T and here must be the A, or here is a A, here must be the T. That is anti-parallel, and thereby they can pair with the one another, and in which here adenine always pair with the thymine, and thymine always pair with adenine with the 2 hydrogen bond only. Similarly, the guanine always pair with the cytosine, and cytosine always pair with the guanine with the 3 hydrogen bond only, and that is also a very important characteristic feature of the this DNA molecule. Then here after the every 10 pairing, here the DNA molecule helically coil. See, after the every 10 pairs. So in other words, what I have to show here is actually is here the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So like this totally 10 I have to write, and very soon here the DNA strand what helically coiled in the right-handed fashion. Helically coiled in the right-handed fashion means in between the the coil, single coil, and there must be the 10 nucleotide. I have written of course here only 5, but the 10 nucleotide must be present, and the distance of this is about 3.4 nm nanometer. The distance of each, the turn from here to here, then again from here to here, and then here to here, and there is a 3.4 nm nanometer, or it is also called as 34 angstrom minute. Remember, 3.4 nm means what? The nanometer, or it is also what? The 34 angstrom minute. So means in this 34 angstrom minute or 3.4 nanometer, how many bases what here depicted? The totally the 10 base pairs. The totally 10 base pairs present in this, and each base pair distance. Each base pair distance. How much? Each base pair distance is about here the 0.34 nm, or it is 3.4 angstrom. Remember this. You may be asking the one more question also. The distance between each helical coil. The distance between each helical coil is about how much? 3.4 nm, 3.4 nm nanometer, or 34 angstrom minute. So here in every this 3.4 nm, totally how many bases present? 10 bases present. So means base pair present means the distance between each base pair is about the 0.34 nm, or it is 3.4 angstrom minute. So he is asked many times for the one marks question, and as well as in the CT examination. So this is also a very important characteristic feature of this. You understood? Then here the nitrogen bases of each strand, the stacks over the another. Nitrogen bases of each strand. So this is stack over this. This is stacks over this. This is stacked over this, because they are anti-parallel. So thereby they may be stacked over one another, and that is also another important characteristic feature of the duplex model of the DNA. And then one more characteristic feature is that here this was the first time explained by Chargaff, and hence it is also called as Chargaff's rule. It is also called as Chargaff's rule. What is this Chargaff's rule? According to the Chargaff, here amount of furin present in DNA is equivalent to the amount of pyrimidine. Amount of furin is equivalent to the amount of the pyrimidine. You understand what I am telling you? So means here, in other words, here in a 100 nitrogen base, or in a DNA strand, nitrogen base pair in a DNA strand. So means here what the 50% will be there. Got it? So amount of furin is equivalent to the amount of pyrimidine. For example, you see, for example, A is a furin, no, and with a double bond with what T is a pyrimidine. No, so means if A is there, T must be there. Similarly, guanine is a furin, triple bond, and it is a C. Cytosine is a pyrimidine. Got it? So means A double bond T, G double bond C. Adenine and this is guanine. Both are the what? Furin bases. So adenine here I have written wrong. Adenine, guanine, and pyrimidine, the thiamine and the cytosine. Wrong. It is written wrong. So adenine, thiamine, and guanine and the cytosine. And so these are the two furin bases, and these two are the pyrimidine bases. If in any DNA molecule here, the total number of furin and the total number of pyrimidine is present is almost equal. See, this is, and here you know what, the adenine is there, correct? Adenine is there, and then the same number is the thiamine. No, then here is the thiamine and the adenine, then thymine, then adenine. So that is why total number of furin is equivalent to what pyrimidine, because in a DNA molecule always a furin combined with the what pyrimidine, or pyrimidine combined with the furin. So that means in a given length of the DNA molecule, the amount of furin is equivalent to the pyrimidine. For example, now one example we will study here. See, in a given DNA, if adenine means it is a adenine is 18%, adenine is 18%, then what is the percentage of adenine is how much? The 18%. What is the percentage of cytosine? See, correct, you read the question. In a given adenine, given DNA, if adenine is 18%, then what is the percentage of the what? Cytosine. How you calculate here? See, adenine 18% means adenine always combined with what? Thymine. Correct? So means adenine 18% means 18 means thymine is also 18, and that is equal to become what? 36. Out of 100, the 36 what percentage is of adenine and thymine. Now if you what minus from 100 minus 36, and you will get how much? The next one is 54, isn't it? Now 54 half is how much? 54 half is 2 to the 4 and 2 to the 4 and it is 27. So means here guanine triple bond cytosine. So means here the guanine 27 is equal to what? Cytosine 27. So then the question is what? What is the percentage of the cytosine? So cytosine is of how much? The 27%. Got it? So means when the adenine is of 18, so adenine is of 18%, adenine if it is 18%, and then the percentage of what? Here the same should be what? So again that is 1 minute. Yes, adenine is 18%, thymine is also 18%, so means 36. So 36 means 54. Yes, correct. So adenine is 18%. So then the cytosine must be how much? The 27%. 1 minute. Yes, so thereby here the 27 percentage of the cytosine is there. So in this way, the Chargaff's rule, according to the Chargaff, here what the total number of what furin and the adenine must be the same. So total number of furin and adenine must be the same, and that should be in the what? The 100%. The 100% ಆಗಬೇಕು. So in this way, what here the amount of furin and the pyrimidine in given in a DNA standard, it always what the equal. That should be the always what the equal. So like this here, adenine, the thymine, then guanine, the cytosine, the number is always the same. So means here the 50% what the furin and then 50% the guanine should be there, and then only it what complete the entire DNA process. So furin and the pyrimidine bases are the same. So in this way, here 18% adenine if it is there, and there must be definitely what the 18% is a thymine. Thymine 18% ಇರಲೇಬೇಕು. So in this way, what the entire the DNA strand completes the process, and in every the 10 base pair it turn and that form a right-handed fashion of the structure, and this is the double helical structure of the DNA, the double helical structure of DNA. And next, central dogma. Next one is the central dogma. What is this central dogma? So central dogma means here in any given cell during the process of metabolic reaction, the DNA play very important role. DNA play very important role, and that to be during here the synthesis of protein and many more metabolic processes in which the DNA molecule act as the main the molecule, and it play also very important role in inheritance of character. And that inheritance of character shared from the one cell to the another cell, or from one generation to the another generation, one cell to the another cell, and from one generation to the another generation. See here, for example, DNA. This is a DNA molecule, and this is DNA molecule. So when it is undergoing for transcription, and that is to be converted into mRNA. You studied already, and this mRNA further undergone for the protein synthesis, and that is protein, and that is during the translation. During what? Translation. So DNA converted into RNA, and that RNA containing one type of message, that is why it is called the mRNA. And this mRNA, when undergoing for the translation, and at that time same is converted into the protein. So means this is the process called as the protein synthesis. So protein synthesis play very, very important role in expression of the gene, and this is also called as a gene expression. And that a gene expression taken place in everybody's cell, and without which here the expression of the gene or synthesis of protein or any synthesis like enzyme, carbohydrate, protein doesn't take place, and thereby our body sometimes suffer from the different type of metabolic process or the metabolic reaction, and then we have a problem. We may suffer from the different problem, and that is what you call as the disease, the defect, etc. starts. So that is why. So DNA acts as a master molecule. And this master molecule have a central dogma, main role. And in this process, in all cell, all living being, a plant, animal, and human being, and it undergone for the transcription, and this transcripted DNA is converted to RNA, and Artha students see this is DNA, and this is a DNA, and this DNA when undergone for a transcription, and that is to be converted into the mRNA. Means the single double stranded structure converted into a single stranded structure, and this is single stranded structure when undergoing for translation, and that is to be converted into here the protein molecule. And this protein molecule is responsible for metabolic reaction, and that may be is in the form of hormone, enzyme, some organic compound, so many more things. All are very important, and that to express the gene. If a protein is formed, then only the expression of gene, otherwise the expression of the gene is not at all possible. So remember, so this is the main role of the DNA, and that is what we call as the central dogma. Next, packaging of the DNA. Packaging of DNA helix. Packaging of DNA helix. See, what is this packaging of DNA helix? See, in our body cell, the chromosomes present. Say, for example, this is a cell, and here is a nucleus, and in this nucleus, the nuclear material is present, and this nuclear material when it is condensed, undergoing for condensation during the division, and same is converted into what? Chromosome. And that you can see particularly in metaphase and the anaphase stage of the division. But when the phase is not there, means the resting phase, and at that time it is just like a what? Nuclear material, or it is also called as a chromatometer. Either the chromatometer or the chromosome, and here the materials are packed, and what material is packed in the chromosome, and that is what? The DNA. So means chromosome is a nothing. Chromosomes is a hereditary vehicle. You must remember that. What are chromosomes? Chromosomes are the hereditary vehicle. Chromosomes are the component of the DNA. Chromosomes are what? Component of the DNA. And this DNA present in the chromosome, and that to be in the two form. That is, one is the genetically active part, and another one is genetically inactive part. So genetically active part of the chromosome is called as the euchromatin. That all you studied, just to go through the notes which is given in the previous class. I am just revising this. And another one is heterochromatin. Heterochromatin. So euchromatin is of what? The genetically active. These are loosely arranged, and they are what? Genetically active. These are genetically active, and they are light in color. And these are heterochromatids are genetically inactive, and dark in color. So means here this is a chromatometer, this is nuclear material. You see this nuclear material, if I'm taken outside, and here somewhere, and there is a dark, means it is like this dark, and then again dark, then again dark. So like this dark is of heterochromatin, this light is of euchromatin. So like this, the dark area and the light area, and in this way, here what? Heterochromatin, euchromatin and the heterochromatin. But heterochromatin, that containing the light region, and that is a genetically active, loosely arranged of this nucleotide of the DNA, and that play a very important role in the what? Here transmission of character from one generation to the another generation. These are the active part, and both heterochromatin and the euchromatin, that form a packaging of the DNA. If you taken out the this chromatin, and that containing so large number of the fibers like structure, and the structural and functional unit of chromosome, and that is called as nucleosome. Structural and functional unit of chromosome is called what? Nucleosome, and that nucleosome is monomer. Nucleosome is a monomer of the chromosome, and that forms the packaging of the DNA. And in the book you studied, see in the book, just go to the book, and in the book you studied like this structure. Now, in the book you studied like this structure. This is a packaging of the DNA molecule. So packaging of what? DNA. See, this is a packaging of DNA molecule, and here this one is H1 histone. This one is H1 histone, and this is a linker DNA. This is linker DNA, and this is octamer. What is this octamer? So that octamer containing the four different type of what? Four different type of histone, but 8 molecules are present. That means particularly the 2H2, 2H2A, then 2H2B, so isn't it? And H2A, H2B. So like this, totally 2 molecules of what? Here 2H2A, 2H2B, 2H2A, H2A and H2B. So like this, totally the 4 molecules of different the histone present, and these histone molecule totally made by 8. So 8, so means here the H1 histone, and then H2, and then what the S3, H4 like this, the histone molecule present, octa means 8. ಇದರಲ್ಲಿ ಟೋಟಲಿ